Join us. I think we're gonna get going. Awesome. Well, just a couple quick housekeeping things. Thank you for coming out to Kings and Wildflowers tonight. I know it's a little wet and it's a little gray. We have a restroom just back there for folks who may have not been here before. We have some cookies, some water, some chips, all that stuff. But yeah, thanks for coming out. We're here tonight to celebrate the work of our TIDALS Fellows who spent 10 weeks studying intertidal habitat and restoration projects on Newtown Creek. My name's Hart Macon. I work for NCA as an environmental educator and got to co-lead the program with my colleague.
I'm Gus. I'm the restoration associate at Newtown Creek Lions.
Nice. So here's how the night's going to go. I'm going to give a little bit of background about the creek, about some of the work that NCA has done on restoration projects using some filter feeders and some grasses. And then Gus is going to introduce the program. And then each fellow is going to present their work. So what their experiment was, their area of interest. And then we'll end the night with some questions and discussion about community science and the work that's being done around the creek. Awesome. We're Newtown Creek Lions. We are a community-based organization that focuses exclusively on Newtown Creek. Our mission is to restore, reveal, and revitalize it. So we have a bunch of different kinds of programming. We do advocacy and work with elected officials. We do educational programming with field trips and kind of open hours in environmental centers. And then we do a lot of like on water and kind of on land restoration work. So that might be like kind of street and upkeep. Sometimes we're doing like intertidal projects, but we're doing a lot all year round. So let's figure out where we are and just get some context. We're right here at Kings and Wildflowers, right next to Newtown Creek on the Brooklyn side. This is Newtown Creek. It's about 4 miles long and it forms a natural border between Brooklyn and Queens. It flows out to the East River. It's a tidal tributary of the East River. So, in our work today, when we go out and look at the creek, it's an industrial canal. It's a place that has been changed a ton. So this is the city that we live in today. Right? It's a city that has bridges and roads and tunnels and delis. New York City has not always looked like this. Newtown Creek has not always looked like this. If we were to go way back, if we were to get in a time machine and look at Newtown Creek like 600 years ago, pre-colonially, it'd look pretty different, right? Does anyone know like what Newtown Creek, kind of the habitat was around like our waterway? I know some folks in the room definitely do.
Should we answer?
Yeah, please. Please.
Marshland.
Yeah, right, salt marsh. Most of the area around our water in the whole harbor was salt marsh, and particularly around Newtown Creek, acres and acres of salt marsh, right? We worked with an artist recently. I work with a lot of students, and we came up with an animation to try and describe what this salt marsh might have looked like. Because when we look out here today, it's kind of looks unchangeable, like it's been here forever, but it hasn't. So this is what, this is an imagining of what the creek used to look like pre-colonially, before its shorelines were hardened, pre-industrial revolution, pre all these changes that shaped our world and our city. Right, so, we're seeing a lot of mud flats. We're seeing a soft shoreline where the tides are coming up, the tides are coming down. Right, it's maybe flowing out to the East River. It's a lot wider back then than it is today. So obviously, that's not what's going on outside our window. We got rid of all the salt marsh around Newtown Creek. We changed the ecology of this place, but it's still a habitat for tons of life. I spend a lot of my life work day now out on the water, and I see tons of animals. So just to kind of like illustrate what this change looked like, this is a bit of a simplification.
I know who did that one.
Yeah, we worked with this artist Wyatt, who came up with this animation.
It's got bug eyes.
Yeah. So our creek is changing in a bunch of ways during the Industrial Revolution. Right? During the 1800s, the creek is deepening, our shorelines are hardening, we're filling in all these wetlands, and the creek is forever changed, right, past the mid-1800s. So we lost a lot of really important habitat. We displaced habitat for animals that have been here for millennia, right? Soft, muddy areas where mussels and oysters and fishing birds can go down and look for food. We changed this place because it was used as a resource. It was used as a resource by corporations that wanted to make money on the creek. It was used by the city that used it to move incredible amounts of material in and out of it all the time. So even though this change took place, it is still a habitat. I see all kinds of life, right? These are some pictures that me and my colleagues have taken down by the creek. These are beautiful water birds that love to hang out on some of the shores of Newtown Creek, right? Big cormorants that are diving down. They're looking for fish, right? They're looking for big schools. We can see a ton of these, right? Up by the surface in the warmer months. We've got toadfish, some of these smaller fin fish that are looking for shelter in the creek, right? Crabs and mussels and oysters. So even though the creek has gone through an unbelievable amount of change, even though the creek has had a history of environmental abuse, neglect, disinvestment, it's still a really important habitat. So our organization, part of our goal and mission is to bring back some of that habitat for these wildlife and find space where they can reclaim it. So this is just an example of some of the projects we've worked on over the years. We've done a lot of surveys to kind of understand existing populations in the creek. So we've done that with oysters, we've done that with mussels. We've recently, like in the past year, we have a kind of pilot partnership with Billion Oyster Project, if folks are familiar with that in the room. It's an organization that is focused on oyster restoration, understanding wild oyster population. So we put some of these cages and concrete bases in Whale Creek, which is a tributary of Newtown Creek, and to try and understand settlement. What's there with the current water quality conditions? What kinds of life can we see? Here's a video of one of those projects. This is one of the members of the, This is one of the members of the Billion Oyster Project staff installing this cage that's filled with blank shell in the hopes that us and BOP will monitor them to see what kinds of settlement there might be. So we're using the existing infrastructure of this steel bulkhead, these I-beams, and placing them down in the intertidal zone. Same with this. So this is a project that Gus got to work on under an old wharf, installing these wooden frames to promote mussel settlement. So we took mussels, seeded them on these wooden frames, and are hoping to monitor them in the upcoming season as more mussels are coming into the creek. We're also working with grasses and different plants. So that salt marsh that used to be here, it was made up of mostly one species called Spartina altiniflora. And so with partners with Sarah Durand and at LaGuardia, we built these living docks a bunch of years ago and continue to work on them to understand kind of the benefit of having some of these smaller scale pilot projects using these native grasses in an industrial waterway. We're working with kind of other organizations. Like there's a group called Seaweed City that's trying to understand how kelp might sequester some of the excess nutrients that's in the water. We did this big kind of replanting in this project that the DEP did a couple years ago, where they had this fresh, new, sandy area put in, and they planted a marsh. And that marsh did well for a bunch of years. And then because of a bunch of different reasons, maybe lack of investment, maybe some problems with geese or elevation, it started to fall away. And so this past spring, we replanted this marsh and are upkeeping it throughout the season. Cool. And I'll hand it over to Gus. So over 10 weeks, we work with these fellows to try and better understand the creek as a habitat. Yeah.
So for background, this is a project funded by the Department of Environmental Conservation. In previous years, we've focused on urban heat island effect. We're joined by a fellow urban heat island fellow today. But this year we are focusing on intertidal habitat restoration. So this is a 10-week program focused on Newtown Creek, very located in this neighborhood. And we started with a couple weeks of field trips to other restoration sites. So working with groups like Jamaica Bay, Rockaway Parks Conservancy, Randall's Island Parks Conservancy, Hudson River Park Project, a lot of different groups, just to kind of learn as much as we can about what's going on in the city in tackling some of these issues around intertidal habitat. Part of this also included maintaining some of these existing projects that Hart showed us before and discussing our research as we're going, right? Discussing how these projects might have a benefit, how they could be improved, what are future opportunities for projects in Newtown Creek. And then a big part of this is science communication for the community, right? So we want to be synthesizing this information and sharing it out with folks like you all who are curious to know more about it, in the hope that we can have more engagement and get more people involved in programs like this. So these are just this is a selfish gallery of photos that we took in our adventures. So we participated in some salt marsh plantings in Jamaica Bay. We did some lab work with Billion Oyster Project, right? So this is part of their community water quality testing program, which has been going on for around 10 years now. This is on Randall's Island. This is in this room with Professor Sarah Duran talking about mussels. Right? And then this is some of our field work that we were doing in the course of our research project. So every fellow has been working for the last several weeks, focusing in on a niche topic of their interest, designing a project, and then collecting all this data in the creek. And for the reasons that Hart laid out, this is a really complicated thing. There's a lot of heat and contamination and pollution and nasty smells to deal with as we're doing this. So I really applaud everyone for how hard they worked and how diligent everyone was in creating this data and creating these projects. Yeah, and without further ado, we're going to get into it. So I want to mention we have 2 fellows who are not with us today who had to go to school early. So we're going to be doing a hybrid in-person presentation and then a pre-recorded presentation. And we're going to start with
Randy.
Yeah, Randy. So Randy, if you don't mind coming up, you can present. All right. All right.
Okay, hi. So my name is Randy. I am a senior, a rising senior at Stony Brook, and I'm here to talk to you guys about plankton within the creek, all about what that's like, what they're about, where they are. What even are plankton, right? So they can be defined as any organism, that be it plant or animal, that on its own cannot move very well. They use water and the currents to basically get pushed from place to place since they lack any real physical ability to do so. And it can be broken down into 2 diverse groups. You've got phytoplankton, which is anything that does photosynthesis, and zooplankton, which is every organism that eats other organisms for nutrients. And one of the kind of important things about those organisms is that they have the ability to bloom, which for the creek is kind of like a major thing. And it happens mostly due to increased nutrients, which causes their population to suddenly go up in the thousands and millions in very short periods of time. You can see it in this picture. This is not the creek, but it is kind of it's along the northeastern coast. And you can see that this is all plankton. This is all just from day to day, they kind of grow in numbers and they kind of can color the water green like that, or sometimes blue. But this is essentially what it would look like at different points. And one of the major contributors as to why that happens is the CSOs that we have along the creek. They are caused by rainfall. So during any rainy events, you'll see that certain sides of the creek or certain locations will start spewing out raw sewage because the system is overfilled and it can't really contain all of it, can't move it all to the right locations. And it's not great for the environment, as you would imagine. It's over 1 billion gallons of raw sewage get pumped out into just our creek alone every single year. So you can imagine that the rest of New York as a whole, it's a lot worse. And you really get to see the effects of that, both on a microscopic scale, but also in terms of just looking outside. The water is not very pretty to look at. And I feel like this is something that should have attention put on it as often as possible. So yeah. This is just a video showing what that looks like. See, it's raining. That's all raw sewage coming out. It's kind of like fecal matter. It's kind of gross. But it happens, and this happens every single time that it rains, since it's about less than half an inch of rain does this. And it occurs pretty frequently. This is also just a map showing where all the CSOs in New York are. We are, or at least the one I was looking at, is up there. That is the Dutch Hills one in that tributary. And it is pumping out on average between 1 and 200 million gallons of sewage every single year, which is incredible. That's a really, really large number for a very small plot of land. And that kind of made it a site that was worth looking at just because of how much is being put into that area so quickly. Which brings me to my question, which was looking at the plankton diversity throughout the creek, mostly focusing on the sites in northern Brooklyn, as well as some along Long Island, especially in Dutch Kills, with a secondary goal trying to connect it back to CSOs, just because they're so prevalent. I feel like it would have been kind of inappropriate almost to not include them. But also trying to connect it back to weather, which would have been really great, but unlike today, it did not rain very much for much of July or much of earlier this month. So I tried my best, but it didn't come all together that way. This is basically showing the route that I was taking, me and a colleague of mine. Again, this is us here. So a lot of it was walking from all of these sites on the bottom and then making our way to a sampling or an analysis lab at Queens Landing Pier. And then on other days, the Queens sites, and then going to the same locations to analyze everything. This is some of the materials that I used. There was kind of like standard sampling analysis and scientific stuff like gloves and pipettes. There's also specific things for this, like the toe in the top left, and also more DIY stuff that I had to come up with on the fly. Like I had to make a weight with the nuts and bolts, which was really interesting, and they got lost, so I pray for them. And I also had to make my own squeeze bottle to help with getting things out of the toe because things can get stuck, and I needed to figure out a good way to do that. There's also this microscope that was provided by the people at Queens Landing, the boathouse, so much appreciated that they let us do that and have access to everything. This is the toe. That is me being a scientist. So generally, you just want to drop it in to wherever you're sampling from to acclimate it to that area, just so that you're not contaminating it from other samples. And after you do that, you bring it up, you attach your sampling bottles, your weights, and you lower it down. And once the bottle is like filling up with your water samples, you have to make sure to bring it up super slowly, which is kind of irritating, but it's to prevent current that you can produce from pulling up the toe too fast from taking the contents from outside the toe back into the water source. But after that point, after you pull everything out, it's pretty straightforward. You cap the bottle after putting everything back inside, and I was able to bring them back to the lab to analyze everything, which was fun. This is a picture of that process, what that looked like. Fancy microscope and everything. I was counting everything by hand through two main methods. One of them was an FOV count, so field of view. I was looking through a microscope at a specific magnification, and I was counting everything by species in one single snapshot of what that was being shown. But I was only doing that when the species was, let's say, too abundant, and you'll count 40 of them in a very tiny image. I would do it then. And if they were less abundant, I was able to do full slide counts, which in retrospect, I should not have done that both ways. It's a learning experience, but I kind of mixed and matched when I felt it appropriate. And this is my results after all the counting and analyzing everything. So we've got the major plankton species on the bottom, as well as the locations for sampling, going from the closest to the Dutch Kills head to the furthest at the mouth of the creek. And also just abundance, and everything is divided by the species as well. What I found most important to take away from this is that at the head of Dutch Kills, where all the CSOs are being pumped out of, in these super short time frames and really high concentrations, you're seeing almost only really bacteria, so cyanobacteria, as well as phytoplankton. That is the grand majority of what's being released and what's being seen in that area. But you're also seeing, you know, lower abundances of things like copepods, which are shrimp-like crustaceans, very small, very interesting to look at, and other kind of worms and larvae that you can see as well. Yeah. Another thing that I felt was really important to take from this is that the numbers, so you'll see that the bacteria is in like the mid 20,000s. That is significantly higher than it needs to be. It is usually anywhere between 5 and 20, but 20 is like a really upper high limit that you shouldn't really be seeing anywhere. Same goes for the phytoplankton and other species of the sort. Just to say that while their growth is being kind of allowed by the CSOs, you never want to see numbers that high or this exaggerated. And this is just kind of bringing that to kind of a further point. You can see that that after scaling everything up to the full length of the creek, as well as just per liter of water that I sampled, you're seeing that some locations per liter are having hundreds of thousands of plankton, and that is not great. It is not ideal that such a tiny volume of water would have such both diverse but also such abundant species in them. The number should be much lower. It is nice to see that they're all there, but to this extent it's not supposed to be that way naturally. Yeah. And this is just me breaking down everything that I saw. So this is just phytoplankton. The top is a species called skeletonema. It's all of that really. And then the bottom is a species called navicula. It's a pennant diatom. That's just the shape that it goes by. And yeah, the top was a bloom. So you'll see that in that one image, that's at like 400 magnification. It's really, really small. And there's like 20 of them in that picture. So scaling that up to a liter of water, you can imagine that there's thousands upon thousands of them, which leads to that coloration that we see in the water pretty often. This is just a video of the skeletonema. You can see that that one on the bottom right is moving. That's because of the water in the slide itself. As I was moving slides around, it would push the water around and would cause whatever I was looking at to move around a bit. But it being stationary is normal. These are all live samples, so this is what they would be like uninterrupted. With copepods, again, these are the shrimp-like crustaceans that you'll see. They are the major zooplankton that you'll find in the water. They're also the major carnivores that you'll find in the water, as well as the major consumers. They're kind of like the biggest group in that bunch. And their abundances were very tied to everything else, where whenever the phytoplankton population went up, so too did theirs. Same thing with the cyanobacteria and things of the sort. So it would explain why at sites like Transmitter Park, everything was really low because the population and the abundance of plankton was really low, of phytoplankton. So these guys couldn't grow and they couldn't spread out very far. Then the last major group that I saw the most of was cyanobacteria. This is basically just photosynthetic bacteria, also counts as a type of plankton. They were by far the most abundant thing that I saw. It also just means that given the nutrient concentration in the water, they had the easiest time growing, and at the same time had the easiest time killing off everything else in the water, because when they bloom, they block out all the sunlight and they sink oxygen really easily just when they die. So plankton couldn't grow and the copepods couldn't grow. Basically everything was at their whim, I'll put it that way. They were pretty important with respect to that. And some other stuff that I saw. So this is a tunicate larva, or a sea squirt. Normally they are like roundish and they don't move a lot. You can squirt them or like press them down and they squeeze water. But their larvae are kind of tadpole shaped. And it really confused me at first because I'm like, why are there tadpoles in the creek? Would have never expected that. But this is what they look like. They're interesting looking. It's got its little eye. It's the one eye on the side. Yeah. This, whenever it appears in the video, is a type of marine worm. Yeah. It is. It's basically the marine equivalent of an earthworm, so they are pretty abundant in the water normally. Another major type of zooplankton that's found. And they move around with They move around with the little hair-like fibers around their head-ish area. It's just interesting to see that they're there. And then this, so much water.
Things are leaking a little bit.
Yes, they are. They are leaking. I can't. Okay. That is just a more mature version of the other one. So you can see that it is much larger, and it's got these horn-like appendages on the front that are used mostly for defense, and they're used to help it move around and push things out of the way. Because it's a larger organism, so there's a lot more like physical debris in the water that it needs help moving. So that's just kind of what it does, what it's there for.
So is this guy going to become one of those clownworms with the...
They can, yes, yeah. So after this whole process, I did enjoy this, do not get me wrong, but microscopy is a little challenging when you're going at it from the perspective of you're doing this for the first time. So, mixing the ways that I was counting things messed with my results in the long run, so I had to go back and change them. Things like not having the appropriate equipment that would have helped sooner. Or just working with New York's weather. I mean, imagine having to do all of this with that happening in the background. It would not be ideal. So, looking forward, if I ever were to do this again, which I probably will, because I do enjoy it, I would definitely simplify the project quite a bit. I would absolutely stick to one type of method for counting. I would pick one plankton group to look at, so I'm not looking at every plant species or every animal species at the same time, just one or the other to make things easier for myself. And yeah, I think that kind of sums up more or less how I'm feeling about it now. I'd like to just take a second to thank everybody for allowing me and my colleagues here to work and learn from you guys for the last couple of weeks. Everybody at NCA, as well as anybody who gave us their time at their locations to teach us about restoration and any of the marshland related work that is being done, because it's stuff that's both applicable to what we're each studying independently, but it's things that we can all put forward and hopefully later on in life make a difference with. So, thank you. Those are sources, whatever. Questions.
I have a question just about what you're, like you said, you're a rising senior at Stony Brook.
Yeah.
But what are you studying, or what, because it seems like a pretty complex thing to do from just being like, oh, I do whatever. Like,
Yeah.
it's already the path you were on.
It is actually. I study marine sciences at Stony Brook, and I recently just took a class almost entirely on phytoplankton. So I felt that I could use what I had just learned and kind of expand on it, grow from it a little bit. It was just really applicable for me. Oh. It's fine.
Because it gave us the idea of the diversity is in the water. And the fact that we're looking at a temporary population of juveniles, the long-term population, then I guess it could come.
Exactly.
And then you have, that are good raisers, and then you have all the single-cell algae. So
Yeah.
it's incredible to me.
It is. I tried to do my best to include as many groups as I could when graphing everything, when showing everything, when explaining. It just came down to what groups kind of provided the most ecological benefit or harm. Some of the blooms are harmful if they grow too much, but a bloom in and of itself isn't necessarily a bad thing, if that makes sense. So it was interesting to try to quantify what was good versus what was bad for the creek, things like that.
Did you see a bloom?
I did not. I really would have wanted to, but I know that had I sampled from English kills instead, it definitely would have been that way. Just because it's much more polluted in that area.
Thanks so much, Randy. I hope people feel deeply immersed in the CSO and wet weather in this room right now. Yeah, this building is really old, and so it's leaking a little bit. Cool. So next we're going to hear from one of the fellows who did work. So she went off to college this week. She her freshman year, but before she left, she recorded her presentation. It is on biodiversity, comparing different shoreline types. So she was out on the water with some of her team members, using a quadrat, taking some samples. Oh, it's really leaking. All right, but we are going to hear from Jackie.
Hello, my name is Jacqueline Bonilla, and I am a freshman at Dartmouth College. I will be majoring in environmental studies, and I am interested in ways that we can sustain ecosystems for the future. This is why I decided to research the effect of shoreline types on biodiversity in Newtown Creek. First, what is biodiversity? Biodiversity is the variety of biological organisms found within a particular habitat. It's determined through species richness and evenness. Species richness is the number of different species or different types of species within that area, while species evenness is the distribution of each of these species in that area. Together, they determine levels of biodiversity. So, for example, low species richness and low species evenness both would cause low biodiversity. On the other hand, higher species richness and high species evenness would lead to high biodiversity. While these two components affect biodiversity, there are still other factors that can influence biodiversity. Some examples are the type of habitat, climate, or environmental disturbances. Over time, Newtown Creek's habitats have changed. There once were native saltmarsh shorelines all around the creek, but through the years, they've been replaced with engineered shorelines. Both natural and engineered shorelines have an area called the intertidal zone that experiences both high tide and low tide. These different conditions allow the shorelines to have a potentially diverse spread of organisms, which increases the ecosystem's overall productivity and success. Bulkheads are a type of engineered shoreline, and they dominate 99% of the creek's shoreline. This map is pretty old, so it doesn't reflect that 99%, but you can still see that bulkheads take up majority of the shorelines around Newtown Creek. And there's very minimal natural shoreline, and which is not even visible on this map. And it would be shown in green if it was visible. So since these bulkheads dominate 99% of the creek's shorelines, this causes people to believe, or it can cause people to believe, that the creek lacks biodiversity. However, bulkhead shorelines differ in materials, which can influence biodiversity. Examples of these different bulkheads used in my research are concrete timber bulkhead, a timber bulkhead, a steel sheet pile bulkhead, and lastly, a stacked stone bulkhead. By analyzing the relationship between shoreline type and biodiversity, we can preserve shorelines that support a greater biodiversity level and improve on the shorelines that have historically harmed the ecological health of Newtown Creek and may weaken biodiversity levels today. So to collect my data for my research, initially samples were going to be taken using the transect method, and the transect method would have a transect line. In this diagram, it's yellow, and this, in my study, this transect line would have ran from the mean high tide line down to the mean low tide line, which is the water level, and any species that cross this transect line would be recorded and sampled, and anything outside of the transect line would not be recorded. And the problem with this sampling method is that it doesn't accurately represent larger shorelines since the sampling area is so small. So instead, for each location, the quadrat sampling method was used, and systematic sampling was also used. For the quadrat sampling method, each quadrat measured 2 feet by 1 foot, and they had strings that ran across them that created 2 by 2 quadrants. These quadrants helped recording data of each species. The systematic sampling was used to select a sampling location without any bias. So I would start from the beginning of the shoreline, and I would measure 16 feet from that start point, and my endpoint would be my sampling location. I did this 3 times per shoreline for each trial. And once I had a sampling location selected, I used the quadrat to collect my data. And I would start from the water level, which was 0.2 feet high, and I would continue taking that quadrat and recording my samples using my phone to take pictures of the species inside or within that quadrat, and I would continue moving the quadrat up until I reached the mean high water level, which was, or mean high tide line, which was 4 to 5 feet high. So once I got my samples, I identified the species I recorded or took pictures of, and I only counted the sessile organisms which sessile means organisms that are immobile or are slow moving, because if they did move, then it would be hard to keep track of them. So this research doesn't accurately show the full biodiversity level of shorelines, but we can get a pretty good idea of what the shorelines have. So once I collected my samples, I used Billion Oyster Project's species identification guide to identify the species within each quadrant. And you can see down in the legend that some of these species were green algae, red algae, clams, mud snails, oysters, mussels, and barnacles, and green striped or orange striped green anemones. So this is one of my bar charts, and this shows the different types of species within each shoreline and the population of each of those species in that shoreline. In other words, the species richness and species evenness in each shoreline. This bar chart also shows the similar idea, or like the same thing, except I divided the invertebrate species from the algae species to give you a better perspective on the diversity of each kind. So using these bar charts, some patterns I noticed was that the steel sheet pile bulkhead consistently had the lowest biodiversity. All the bulkheads had five different species identified overall among them, except even though they all had five species, they all didn't accurately, or they all didn't have the same species evenness. We can see that the steel sheet pile bulkhead, you can only see three major species from the bar chart, which implies that there is very low species evenness among, like, within that bulkhead. And this very low species evenness decreases the biodiversity of that shoreline, and it's the lowest among all the shorelines. Contrary, the concrete timber bulkhead had the highest biodiversity um, among the bulkheads, but also among the other shorelines. We can see that there is pretty good evenness between the anemone, ridden mussel, and barnacles. So that high species evenness causes it to have a higher biodiversity. And the concrete timber bulkhead didn't have the highest species richness though. That goes to the natural sloping shoreline, which had 6 different species identified within its shoreline, causing it to have the second highest biodiversity. The reason why it's the second highest is because if we look at the bar chart or the bar for natural sloping shoreline, it has very low species evenness. And this low species evenness contrasts the high species richness, which lowers the biodiversity level. So looking at these patterns, we start to wonder what causes shorelines to have higher biodiversity than others. And a potential answer for this is the materials and textures used. With the steel sheet pile bulkhead, the surface was very flat and was smooth, which can make it difficult for some organisms to attach to, and it didn't have much depth to its surface. So this could have decreased the species evenness since many organisms couldn't attach to its surface, or like it wasn't the ideal surface for them. On the other hand, the concrete timber bulkhead was made of 2 different mediums, concrete and timber. And concrete, it's rough. It has like, it doesn't have a smooth surface. And the timber, it has like cracks in it, which both of those can give additional habitat to organisms, which can be a potential reasoning for why there was higher species evenness among the species in that shoreline. Lastly, the natural sloping shoreline had the greatest variety of materials used. We can see it in this image now that it has natural sediment and it has different size rocks across its surface. Oh, and another thing is that it sloped. So it was the longest shoreline sampled, and this extra distance also provides more habitat. There's also like more plants around its shoreline, and some of that vegetation, like some leaves were found across the shoreline, and this also provides additional textures and materials that can serve as habitats for the organisms on that shoreline, which can explain why it had the highest species richness or highest diversity of species, as it had the greatest variety of materials and could provide different types of habitats. So what this implies is that using eco-friendly materials and using a variety of these materials can add to the quality of the environment and improve the ecosystem for that shoreline, leading to higher biodiversity. And if more shorelines had greater variety of habitats available and better eco-friendly materials were used for it, then this can significantly improve the overall biodiversity of Newtown Creek. These are my works cited, and thank you for listening, and thank you to the Department of Environmental Conservation and to the members of Newtown Creek Alliance.
So Jackie is at Notre Dame, and she's the youngest of our group, and she worked really hard to be out on the water. She went out on the boat, she went out on some of these shorelines that are a little unstable, took many pictures, spent a lot of time analyzing data, and put this together. If folks want to look closer at her data and all the fellows' data, there's a poster in the back. Yeah.
Really quickly, it's raining a little bit.
What?
I know, and they were so engrossed in the presentations. I just want to note that there was like a little bit of water coming into the elevator shaft. So I think it would be advisable to avoid using the elevator. And then as we're walking out, there's a couple like patches of water on the stairs. So let's be really mindful about not sitting in water.
Awesome.
And I'm also going to put this towel right here.
Thanks, guys.
Yeah.
Do you want to put it back there?
Yeah.
Sorry.
Would you mind?
Yeah.
Oh man.
Cool. Okay. Next, we're going to hear from some folks in the room. We're going to hear from Tiffany and Crystal, who worked on a project to focus on the rib mussel and how that rib mussel reacts to other environmental factors. Nice. Cool.
Hello everyone, my name is Tiffany and I'm a rising senior at Stony Brook University majoring in environmental studies.
Hello everyone, my name is Crystal Vasquez. I am a student at LaGuardia Community College and I am majoring in environmental science.
So our research project is on the effect of Spartina alterniflora on rib mussel clustering patterns.
The Newtown Creek was once a thriving waterway and has now been heavily altered by centuries of industrial activity. Its natural landscape has significantly changed due to pollution, shoreline hardening, and constant dredging. Despite all this, rib mussels have found their way to still attaching themselves into the shoreline, and they can attach themselves to any kind of surface. So like pilings, rocks, or even themselves. This is an image of a survey that was done by NCA. It's the population of rib mussels within Newtown Creek.
So what are rib mussels? So they are a native bivalve species to the Atlantic coast of North America, and they are filter feeders who can filter up to 30 gallons of water a day, moving particulate matter, harmful algae, excess nitrogen, and fecal bacteria such as E. coli and Enterococcus that comes from us. Yeah. So using byssal threads, which are strong silky fibers made from proteins produced by their foot, they can anchor themselves onto surfaces and attach to different kind of substrates mentioned, such as like rock, wood, and to other rib mussel shells as well, as they provide a good textured surface. And this is a collection of photos collected by NCA, and you can just see different rib mussel habitats found around Newtown Creek. So like small crevices among big rocks and on like timber wood, like in between the spaces of it. And if you remember what Jackie mentioned, like a typical steel bulkhead on the top left here would not provide any like textured surfaces for rib mussels to attach on. So everything else here, you can see the abundance of rib mussels.
So what is Spartina alterniflora? Spartina is also known as cordgrass. It is a native salt marsh that was abundant around the creek. This plant covered around 1,200 acres of wetlands, and it helps improve water quality and also absorbs excess nutrients and produces oxygen. Spartina colonies often grow in continuous parallel bands around shorelines. It also prevents erosion and helps stabilize sediment. This image is a small glimpse of what the shores of Newtown Creek used to look like. Nearly all of the shorelines now are bulkheads.
So rib mussels and Spartina actually display a relationship called mutualism. So mutualism is a relationship when two species benefit each other by interacting with each other. So based on a prior research study that kind of inspired our research project, it was found that Spartina display chemical cues when in contact with rib mussels, and rib mussel larvae was settled and metamorphosed at a higher rate in the presence of the Spartina grass. So you can imagine that rib mussels grow better in the presence of Spartina grass, basically. So how do they display the mutualism? So mussels can attach and form clusters at the base of the Spartina grass stem, as you can see here from a glimpse of our experiment. And clusters are basically just like multiple rib mussels, like all together, just like a little ball. and they use byssal threads to attach to one another, and it's actually like really strong. Like if you try to pull them apart, there's gonna be like some kind of like force there. I don't know. Yeah. So mussels deposit nitrogen-rich fecal material on the settlement surrounding the Spartina, and the fecal material is called feces and pseudofeces. So even though it's excreted by mussels, they actually provide a lot of benefit to the Spartina grass, allowing like nutrient for the Spartina to grow. And because the mussel clusters are so tightly knitted together and like they're so strong, it prevents the Spartina grass from erosion and allows them to grow tall and upright. And on the other hand, the Spartina provides the mussels a good stable habitat and protects them from different predators like blue crabs, and etc. So our research question is to explore whether the presence of the Spartina alterniflora grass can contribute to rib mussel clustering patterns using two different indicators. So we will look at the number of clusters formed and how many mussels are in each cluster. So basically like the cluster size.
Goals of our experiment. The goal of our project was to deepen our understanding of the mutualistic relationship between Spartina and mussels, but specifically we're asking if Spartina influences how rib mussels start clustering. By exploring this interaction, we also hope to generate insights and inform future restoration efforts, not just here at the creek, but New York City and other waterways. So some of the materials we were using for this experiment were 2 aquarium tanks. We also utilized mussels from the Living Dock at North Henry Street. What was really helpful to monitor their environment was the YSI, which is a conductivity meter. So it helped us look for dissolved oxygen, temperature, and salinity to see that the mussels were in their proper ideal environment. We also use reef phytoplankton to feed the mussels. We use this vernier caliper to measure the mussels. We use 30 millimeters to 70 millimeter size mussels. We also used a trail camera for our third trial, and a siphon for water transport. This experiment was a tank-based project, and we held it over 3 weeks. So each week was a trial of 4 days, and as you can see here, our control tank was tank number 2. Our first tank had 15 plexus for time alternate flora, and we arranged mussels to be in the 5x6 grid. We also placed play sand at the bottom of each tank. It was approximately 2 inches of sand, and each tank had an oxygen bubbler to keep dissolved oxygen levels good. Regarding the environment of mussels, as I just mentioned, we had daily measurements of what I just said, dissolved oxygen, temperature, and salinity. And for observations, we took photographs every day. So from Tuesdays to Fridays, we had visual observations. And for our third trial, we used the trail camera to observe movement. Also, I want to mention that each trial had no variable. Like, we didn't change any variables for each trial. It was the same experiment for all of the trials. Every time we held a new trial, we cleaned the tanks, we adjusted and replenished the sand if it was displaced or something, and mussels were fed 2 to 3 times a week, 6 milliliters.
So we, using the trail camera, we were able to put together like a time lapse of our third trial. So we wanted to show mussel movement. So here it is. You could see the 5x6 grid that was placed initially, and then just like some of it. This is like, if you see like a yellow long thing coming out, it's like their foot, which is what they use to move. And yeah, and then this is nighttime. They're just like opening up and closing.
Gapping.
Yeah, called gapping. And you could kind of see that they're already out of the 5x6 grid, going into their respective clusters. Yeah, and then morning comes and boom, like like they're all in like together. Yeah.
Attaching to the glass.
Yeah. And yeah, that's a nice short video. Oh. Okay, so if you imagine, if you imagine all these individual mussels as coordinate points, this would look like what I'm about to show. So basically, these 2 pictures are pictures of Friday, which is the last day of each trial. And then using the last day of each trial, from each trial, we were able to produce these graphs. So you can imagine the x-axis as the length of the tank and the y as the width of the tank. And each little circle is a mussel. So row 1 over here is tank 1, which is the tank with the grass, and row 2 is the tank without the grass. And each column is trials 1 to 3 respectively. So it's kind of hard to tell here with all the different colors, but row 1, which are the tanks with the grass, have a greater number of colors, which means that they have more clusters defined. And the second row, there are less colors, which means that there are less clusters. So in order to put these individual mussels into coordinate points, we use a software called ImageJ. So you can imagine we're tracing each individual muscle of the photo of the muscles, and it produces like a center coordinate point, and these are all the center points of a muscle. And using a method called k-means clustering, we were able to basically visualize all these clusters into their respective colors or different clusters. And you can also see that the clusters in the tanks without Spartina, even though there are less colors, but there are greater amount of points in each cluster. Whereas in tank 1, the clusters there's more distinct colors, but each color has less of its own points, if that makes sense. Yeah. And to kind of put all of that into numbers, the green frequency bar graph is representing all the clusters from all the trials of the tank with Spartina, and the brown bar graph represents all the clusters in the tanks without Spartina. And you can kind of see like the green bar graph is like has like a left shift, whereas the brown one is kind of more in the middle or like to the right. So basically what that means is that more than half of the clusters of the tanks in the tanks with the Spartina grass had a size of 2 to 3 muscles, which is pretty small compared to the sizes seen in the tanks without the grass. So in the clusters found in the tanks without the grass, half of the clusters had a size of 6 or more muscles. So it's like 2 to 3 muscles per cluster, and then 6 or more muscles per clusters of the Spartina tank and like without the Spartina tank. So some general results include that. So the muscle clusters were seen to form within a day. So from that time lapse, that is within 24 hours of the trial, and they have already produced spissle threads attaching to one another. They also attached to some of the sand particles that were more like bigger, like rock kind of, and they also even attached to the glass tank at the bottom of the sand, which was kind of surprising to see since they had to like dig all the way down and then produce the thread to attach to the tank. In this photo right here, you can see one rib muscles attached to the base of the Spartina plant. Like, even though it was just one, but compared to, I mean, like, with the options it has, like attaching to the glass, attaching to the sand, or other rib muscles, it chose to attach to the Spartina base, which was really cool. Yeah. And briefly summarizing, in the tank with the Spartina grass, there are more different clusters, more different balls of muscles, but the size of each cluster were smaller. So there were 2 to 3 muscles per cluster. So what do the results mean? So based on our findings, we think that the presence of Spartina may promote a wider range of settlements. So the clusters will cover a wider range in the tank compared to the tanks without the Spartina. So a broader distribution also can be related to increased ecological benefits. So some of the natural ecological benefits that rib mussels do, they're like natural filter feeders, and they help stabilize settlement. So with them being on a broader space, they can stabilize settlement across a wider range of area, and they can also cover a lot more footprint in filtrating water.
Well, as any other experiment, we also had some challenges doing the experiment. Even though this experiment did provide valuable insight, there were limitations, and the first thing was that the tanks didn't replicate the natural environment of rib mussels. They're always exposed to an intertidal cycle, and this was not the case. They were always exposed to water, and this might have influenced their clustering behavior. We were also limited by the sizes of the tanks, the amount of mussels. So if we would to do this project in the future, it would be helpful to have a larger scale project to see if Spartina is actually influencing how mussels cluster.
So kind of what just Crystal mentioned, but in the future it will be helpful for longer term observations of rib mussel clustering patterns and field-based experiments, like seeing, tracking and looking at the mussels out in the field with the Spartina grass in a natural environment where there's always like variability. But in our tanks, of course, they're always kind of in the same condition. Yeah, and we also hope that there's a better tracking technique or data analysis for the mussels, because even though we did put them into coordinate points, which is helpful to visually see them on like a scatter plot, but it didn't truly replicate the size of a mussel on a plot. They were just like the center point of the mussel, so sometimes we weren't able to see them. We weren't able to see some of the points as close as we wanted to, because they were just the center point of the mussel. Here are our references. And special thanks to Gus, Hart, Willis, Priscilla, Sarah, Dr. Zarnock, and Newtown Creek Alliance for providing us all the resources, insight, and the space, and opportunity to do this research
Yeah,
project.
this project wouldn't have been possible without you guys.
Yeah. Oh, question?
Sorry, I have a question about the scatter plot.
Yes.
So you said the different colors represent different clusters, but what does it mean when there are multiple points that are the same color? Are those like individual mussels?
Oh yeah. So basically the method that we use to put them into colors, basically each respective color was designated as a cluster by the method that we use. So, because if you were to start with just all the points if you were to start off with all the points being just one color, let's say that would just mean one cluster. But we visually identified how many clusters there were at first, and then the system would put them into respective colors depending on like the distance. It's called I forgot what it's called, but it's like a
K-means, right?
huh?
K-means.
Yeah, like K means
So
cluster.
is this one of your experiments, or is this over time like all of your experiments, and that's what that's the data you fed it?
Yeah, so if you think the row 1, each plot from left to right is a different Friday, because each Friday we take like a photo of the ending cluster. So this will be Friday of trial 1, Friday of trial 2, and Friday of trial 3 for tank 1. And then the bottom is just tank 2.
So when you have like the blue, or there are a few blues, like the green,
Yeah.
there are more green dots, there are more individual mussels in that photo.
Yes.
Okay.
Yeah.
Gotcha. Thank you.
So each dot represents a muscle?
One muscle, yeah.
Mm-hmm.
And then you just decide by virtue of it, like some distance that you decide, if they're that close, then that can be considered a cluster.
Yeah.
And so then, and instead of having cluster A, cluster B, cluster C, you have the colors.
Yes.
You could hypothesize that another mussel becomes more attractive in the absence of Spartina and in the presence of Spartina.
Yeah.
They are less attractive, and therefore it's not, there's only one kind to hold on to, I'm going. But with the more natural chemistry offered by the plants, you have a greater distribution.
Yeah, like more even distribution across the space.
So you're tracing movement across each week, or was it reset and we're just seeing each trial as separate, like distinct from the other trial?
Yeah, so at the end of each trial of each week, which is Friday, we basically reset them again. So they start off with the 5x6 clean grid, and then at the end of each trial, we see the clusters again.
So the condition for the top row was consistent, well, at least what you're doing, like the independent variables, and
Yeah.
same thing for the bottom, just two different conditions
Yeah.
done 3 times.
Yeah.
Okay. And what kind of like inference can you draw from the different results, because they look like placed different places? But are there any like similarities that you were able to analyze in terms of the clustering?
Well, I mean, generally, like, if you see, like, let's say this is the tank with the Spartina grass, you kind of see them, like, all around. Whereas here, the clusters are, they kind of just, like, and they have, like, so much space between them compared to a more scattered area.
It feels like the bottom row is much more like distinct groups. Like,
Yeah.
like, intuitively, it feels like, oh yeah, that would be a group.
Yeah.
But for the top ones, there's like a lot of different cases where I'm like for example, the orange and the green, you know,
Yeah.
that's so strange how
Oh, yeah.
that would go with orange. So I was curious if there's any trends that you noticed, like within one condition, in terms of the movement, you know, because it looks like it moved. I mean, I can't tell anything. I don't know how it moved, but it does end up in different positions. I'm curious, like, what's the reason for that?
Yeah. Well, if there was a reason, that would just be the presence of the Spartina grass. So, yeah.
Why are they so attracted to the right side of the tank?
Oh,
yeah.
Is there current?
No, if you imagine, they all start from the right side of the tank already, because this empty space here compensates for the space where the Spartina grass is plugged.
I see.
Yeah, and then this over here, we compensated that same area by placing the mussels more to the right when the trial started.
Awesome. Thank you,
Yeah.
guys.
Thank you.
Right. Yeah. Okay, so we're going to finish up with one last presentation from a fellow who's not here, who's actually on a flight right now to go back to school in Scotland at St. Andrews. He's a sophomore there in the sciences. So I'm going to pull up his presentation. His name's Alex. He grew up in the neighborhood, and his research project is focused on looking at microplastics all across the creek.
Hello, everyone. Thank you so much for coming. Thank you so much for watching. My name is Alex. I'm a rising sophomore at the University of St. Andrews, and I'm really excited to talk to you guys here today about microplastic hotspots on the Newtown Creek. So a little background to my project and a background about microplastics. So what are microplastics? Microplastics is any piece of plastic less than 5 millimeters in length. That's about the length of an eraser on the back of a pencil. These can be degraded pieces of clothing, degraded pieces of kind of household wear, degraded pieces of pellets that are used in industrial production while making larger things out of plastic. And these plastics end up in our water bodies through runoff, but primarily through combined sewer outflow. But what is combined sewer overflow? So combined sewer overflow can trigger for as little as 1/10 of an inch of rain. So this is if rain falls onto the street, it overloads our sewer systems, it overloads our water processing, the city's water processing, and it gets dumped into our water bodies. And in our case, the creek. So CSOs in the creek, there are 450 CSO pipes in the city. 22 of those are on the Newtown Creek, where we are here today. So that's 1.2 billion gallons of combined sewer overflow water that gets dumped into the Newtown Creek. And 200 million of this is in the Dutch Kills tributary. So that's up here, and that's what I'm going to be focusing on for my project. So my question is, does the presence of combined sewer overflow or combined sewer outflows correlate to a higher density of microplastic fibers? So for my methodology, I needed a bunch of materials. First, we have the gizmo. This, courtesy of the Billion Oyster Project, is a device with a weight, a clip, and a rope attached to it for me to dunk my sampling bottles off of bridges, off of like the street ends where I'd be sampling from, to collect water while keeping myself safe. And of course, I needed the polyvinyl chloride gloves. These are marine safe. These not only are microplastics in our combined sewer outflow, but also just untreated sewage, bacteria, and I really need to keep myself safe with as much PPE as possible while doing this project. I had coffee filters and the binocular compound microscope and slides for my project, which I'll work through in my methodology. But here's my route, focusing on the Dutch Kills CSO cluster. So if you're live in person, we're here right now at Kingsland Wildflower. This is where I was working mostly. And then I processed my samples up here at the Queen's Landing Boathouse Marine Lab. So I took 6 samples today. I would go to the Nature Walk. I would go to the Manhattan Avenue street end, go to Newtown Barge Playground, WNYC Transmitter Park, and then Borden Avenue Bridge in Queens, and then the Hunters Point Avenue Bridge. So I would walk from Kingsland Wildflowers along the creek towards the East River, then from here cross into Queens, get the 2 sampling sites on the Dutch Kills tributary, and then go to the boathouse. So I chose these sites for very specific reasons. I wanted to get as much from the tributary as possible, from the Dutch Kills tributary, where there's this huge cluster of combined sewer outflow pipes. And then I wanted to collect samples as far from that as possible. So WNYC Transmitter Park is in the middle of the East River. So that was going to act as my control sample of what water is like, what microplastics are in the waters in the rest of the city's water bodies compared to the very specific tributary that I was working on. So here's my process. I sampled twice a week for 3 weeks. So 36 samples in total, given that I was getting 6 sampling sites a day. I would pass these samples through a coffee filter. Then I would pick up the residue with clear packing tape, which I would line onto my microscope slide and analyze at 40 times magnification. I'd take a 1 centimeter in diameter circle and record the total number of and color of fibers that I found. But as with any project, there were many challenges. My largest one was this limited data set. I only ended up taking 36 samples because this was just a summer research project. I didn't have the time or resources to collect as many samples as might have been better. Materials and resources. We started out with a less powerful microscope. We lacked some of the materials that kind of do what would typically be done for this kind of project. So with more funding, with more resources, there's always more accurate samples that could be taken. And finally, sampling conditions. The Newtown Creek and Greenpoint, Maspeth, the North Brooklyn Industrial Business Zone is considered the hottest area in New York City, one of the hottest areas in New York City. And so sampling was rough, especially given on these kind of 95 to 100-degree days. Going on these long sampling runs was hard. Finally, let's break down our data. So as you can see here, there is a loose correlation between the location of the CSO pipes and the total density of microfibers. All the way on the left here would be where our CSO pipe cluster would be at the Dutch Kills tributary. And all the way on the right would be the East River. And as you can see, this line of best fit, there is a higher density of microfibers closer to the CSO pipe than at the creek. And then at the East River. So now, kind of looking at this data in a different way, here's it mapped out using ArcGIS. You can see a huge quantity closest to the tributary, with the highest being where the tributary meets the rest of the creek, and a very low density out in the middle of the East River. So you can kind of follow the path of these microplastics, kind of going this way, down the creek, and into the river, slowly getting mixed with the rest of the body. One of the reasons for this is something called tidal exchange. The East River's waters get cycled twice a day constantly, while the Dutch Kills tributary in the creek has very low tidal exchange. meaning that waters exchange throughout that water body very, very slowly in comparison to the rest of the East River. There's less time for water to circulate, less time for these microplastics to leave that area, leading to higher accumulation, larger bioaccumulation. Now, one of the important things about this map is that this is including clear microplastics. I found more than 80 times more clear microplastics than any other color. So if I remove these clear microplastics from my graph, this is only what colored microplastics would look like. That's primarily black, blue, yellow, and red. If you can see, there's a huge quantity of colored microplastics closest to the Dutch Kills tributary, and definitely tapering off as you go into the river. Now, I've got to admit, I don't totally understand this data. Why is there so little? Why are there so little colored microplastics at Borden Avenue Bridge? I'm not really sure. It's only about a 4-minute walk between Borden Avenue Bridge and Hunters Point Avenue Bridge. What happens in between those two sites, leading to this dramatic fall off? Also, there's only red colored microplastics inside the Newtown Creek itself. Why is that? Why are there no yellow microplastics found at Hunters Point Avenue Bridge, where I found the highest number of microplastics? A lot of these questions I can't answer with my limited data, but hopefully with more data, with more analysis, we can come to these conclusions. In the end, I was really hoping to try and find a correlation between rainfall triggering these CSO events and microplastic density. And unfortunately, I was unable to find a correlation. With a brief disclaimer that I didn't take enough data. More data gives more accurate results, and I would have been more confident in saying that there was no correlation. But this water here, this line here represents the precipitation taken at in Hunters Point, and these graphs represent the total number of microplastics that I found on each of those dates, with the colors representing the various locations. And as you can see, there's very little correlation. But with more data, I hope to kind of process this better and get a better results. So in conclusion, what did we find? We learned that microplastics are in our waters. Microplastics are linked to kidney failure, endocrine disruption, and certain types of cancers. So we know this is an issue that New York City has to deal with and has dealt with. And this project kind of just solidifies that this is a problem. Here's one of the colored microplastics that I found. This was a red piece that I found at Borden Avenue Bridge. But my project has come together to say that there does appear to be a greater density of microplastic closer to these combined sewer outflow clusters. These pipes, there is more microplastics found in the Dutch Kills tributary than out in the river. So how can I improve this for the future? This is one of those clear microplastics that I was talking about. I found so many more of. There is more to learn. This was a summer project. I only worked on this for the 10 weeks of the internship. I was only focusing on sampling for about 3 or 4 weeks. So in the end, I can learn more, and we can all learn more about what microplastics are, how they can affect humans, and maybe why there's such a high density inside of the creek and inside of the river itself. Another big issue that we had was that there was no standard scientific methodology for collecting these microplastics. We put a lot of effort into working with the Randall's Island Conservancy with kind of developing a smaller methodology of what organizations like us might be able to do. But with a standardized procedure, we can kind of keep data sets aligned across multiple fields, multiple testing sample sites. And so that's a real improvement that can be made in the sense. And of course, we need more data. I only took 36 samples, 6 sampling sites, 6 days, more data, more accurate results, more correlation. But in the end, thank you so much all for coming. I hope that you found this project entertaining and informative. I had a lot of fun doing the research, and I'm really proud of the results I was able to throw together. And I hope you guys see the same correlations that I did. Here's all of the research that I did. Special thanks to the Billion Oyster Project for lending us their gizmo, and the Randall's Island Conservancy for helping us out in learning the methodology for doing this simple microplastic processing. Thank you to everyone who helped me with pictures and with making this presentation. Hope you guys have a great rest of your day, and thank you so much for watching.